the genome does not in any way, and this is not a controversial thing, this is a fact, although people don't emphasize it enough, the DNA in no way codes for the actual form and function of your body. What's in the genome is not a description of the organs you're going to have, how those organs are arranged relative to each other, how they grow, what they do. These things are not directly in the genome. If you actually read the genome, what you see is protein sequences. You see a description of how to make the tiniest level hardware that every cell gets to have, all these little proteins. Now, that hardware is amazing because when every cell has that correct hardware, they are able to do all of the goal-directed computations that allow them, first of all, to maintain cellular kinds of activities, but more importantly, to work together as a collective intelligence that then ends up building the various organs in the body and so on. And so there is a blueprint, but that blueprint arises from the physiology. It arises in the computations of that collective intelligence of cells. That blueprint is not directly written in the DNA. I'm going to do it so that instead of this one head, one tail electrical pattern, you have a two-headed pattern, right? You're just editing the electrical information in the network. When you do that, guess what the cells build? They build a two-headed worm. And the coolest thing about it, now no genetic changes, so we haven't touched the genome. The genome is totally wild type. But the amazing thing about it is that when you take these two-headed animals and you cut them into pieces again, some of those pieces will continue to make two-headed animals. So that information, that memory, that electrical circuit, not only does it hold the information for how many heads, not only does it use that information to tell the cells what to do to regenerate, but it stores it. Once you've reset it, it keeps. And we can go back. We can take a two-headed animal and put it back to one-headed. So now imagine, so there's a couple of interesting things here that have implications for understanding of genomes and things like that. Imagine I take this two-headed animal, oh, and by the way, when they reproduce, when they tear themselves in half, you still get two-headed animals. So imagine I take them and I throw them in the Charles River over here. So 100 years later, some scientists come along and they scoop up some samples and they go, oh, there's a single-headed form and a two-headed form. Wow, a speciation event. Cool. Let's sequence the genome and see what happened. The genomes are identical. There's nothing wrong with the genome. So if you ask the question, so this goes back to your very first question, is where do body plans come from, right? How does the planarian know how many heads it's supposed to have? Now, it's interesting because you could say DNA, but what as it turns out, the DNA produces a piece of hardware that by default says one head. The way that when you turn on a calculator, by default, it says 0 every single time, right? When you turn it on, it just says 0. But it's a programmable calculator, as it turns out. So once you've changed that, next time it won't say 0; it'll say something else. And the same thing here. So you can make one-headed, two-headed, you can make no-headed worms. We've done some other things along these lines, some other really weird constructs. So this question of, right, so again, it's really important. The hardware software distinction is really important because the hardware is essential, because without proper hardware, you're never going to get to the right physiology of having that memory. But once you have it, it doesn't fully determine what the information is going to be. You can have other information in there, and it's reprogrammable by us, by bacteria, by various parasites, probably, things like that. So the hardware-software analogy is actually pretty good here, although a lot of people push back on thinking about living things as computers, and that's because today's computers don't capture most of what's interesting about biology. I mean, that's true. But the thing that is good about this computer analogy is that it allows us to very carefully separate what you know about the hardware. And so if you have a computer and you wanted to debug a program, or you wanted to get it to do something good, would you be working at the level of the copper and the silicon? I mean, you might if you were a CPU designer, and there are certainly people who do that, but the vast majority of powerful things you can do with a computer don't take place by altering the properties of the copper and the silicon and the aluminum and everything else that's in there. They take place at the level of reprogramming and taking advantage of the native intelligence of the machine, the interface that it gives you to control the whole. You know, it's the algorithm that makes the electrons in the machine dance to a particular computation. So my claim is just very simple, that for certain kind of low-hanging fruit, single gene diseases and maybe some cancers that are caused by very specific mutation, it may be possible to address that on the genetic level, maybe. But for the vast majority of what we want from regenerative medicine, we shouldn't be focusing on the hardware any more than we do that in our information technology. We should be focusing on the native intelligence of the cells and tissues and on communicating with that intelligence to push it towards specific behaviors like building a correct organ, maintaining us against aging, and so on. So this is how I see the problem. We really are very bad at recognizing intelligence in unconventional embodiments, where our basic expectations strain against this idea that there could be intelligence in something extremely small or extremely large. People often criticize this approach by saying, well, then anything goes. If you can pick up a rock and say, I think this rock is cognitive and intelligent, there's a spirit with hopes and dreams inside of every rock. That's not what this is. This is quite different. As an engineer, what you have to do is you have to come up with a way to look at a particular system that doesn't overestimate its intelligence or underestimate its intelligence. If you treat complex animals as if they were clockwork, you're going to miss everything that's important and exciting about how they work. If you treat a clock as if it had a complex intelligence, you're going to waste a lot of time. But getting it right is fundamentally important. And so you have this spectrum where the engineer has to pick the right level for the right system. And it's critical not to then say, well, that doesn't sound like human intelligence. Right, we're looking for the basic minimal version. So I think it should have two things. The first thing it should have is some degree of goal-directedness, some ability to take different paths to get to the same goal. So this is William James's definition of intelligence. And it has to have some ability to undertake actions that are not completely determined by local circumstances. So you start with all of that in systems that have scaled up those basic, very fundamental, non-zero levels of agency. We call that life. So it is a spectrum, and I think that in this universe, it goes all the way to the bottom. And of course, they often work in other spaces. So we're good with three-dimensional space, but imagine if we had a primary sense of our own blood chemistry. If you could feel your blood chemistry the way that you currently see and smell and taste things that are around you, I think we would have absolutely no problem having an intuitive understanding of physiological state space, the way we do for three-dimensional space. I think that we would immediately be able to recognize our various internal organs as intelligent agents navigating that space and solving these kind of problems, all the various things that happen during the day. And so here it's a tick, but you can think about a bacterium or something, and it has a little bit of it has the cognitive light cone is quite small because it has a little memory going backwards and has a little bit of predictive power going forwards in time. But what it really cares about is the local concentration of whatever molecules it cares about right at that local level. So it's very small. The cognitive light cone is very small. And you can move up to something like a dog. So that has a longer light cone going backwards. It has a little bit of more predictive capacity going forwards. And of course, spatially, it will know things like one of the goals is to keep intruders out of its neighborhood, the perimeter of the house or whatever it is that's what it works on. But so that's great. That's way bigger than the tick, but or the bacterium. But what you're never going to get your dog to do, as far as we know, is to care about what happens in the town 3 miles over a month from now. It's just not going to happen. They're not capable of it as far as we know. And then you have very large light cones like humans that may really be, one of the goals may be something to do with world peace and what's going to happen. Some people are literally depressed because the sun's going to burn out, and so they're working on these technologies to get us off the planet and so on. So you could and of course we all have limitations, right? So even though the human light cone is huge, and it does have this interesting feature, maybe the first one in this evolutionary chain, maybe not, where some of those goals are guaranteed not attainable. Yeah. So if you're a goldfish and you have some goals that reach, let's say, a half an hour forward, that's absolutely attainable. You're very likely to live that long. And so all of your goals are attainable. If you're a human, it's very likely that that many of your goals are your cognitive light cone is longer than your lifespan, you know they're not attainable, and that may or may not result in some psychological pressures that are unique. But I don't know. We took tadpoles that normally do not regenerate. There's a stage at which they can't regenerate their tails, and we developed a bioelectric cocktail that induces it to grow. My postdoc at the time, Kelly Chang, said, I soaked them, and I said, well, how long did you soak them for? And she said, an hour. And I thought, but that's got to be too short. There's no way an hour soak is going to do anything. And sure enough, that hour soak led to 8 days of regeneration where we don't touch it at all. And the most recent version of that work is in the frog leg, where we show that 24-hour stimulation with our cocktail induces a year and a half of leg growth, during which time we don't touch it at all. So the amazing thing there is, again, this is not micromanagement. This is not 3D printing. This is not us telling every cell where to go during this incredible year and a half long process. This is at the very earliest moment you communicate to the cells, go down the leg building path, not the scarring path, and that's it. And then you take your hands off. It's calling a subroutine. It's modularity. It's relying on the competency of the material, where you're not going to micromanage it. So that was the first time that kind of became obvious that was possible, is when she showed that just an hour stimulation of the correct bioelectrical state got the whole tail to commit to regenerate itself. I really wanted to work on cancer, and I really wanted to work on this idea that there's a bioelectric component to it, and the way you can think about it is simply that not why is there cancer, but why is there anything but cancer? So why do cells ever cooperate instead of being amoebas? Why do they ever cooperate? And so we know that the bioelectric signaling is the kind of cognitive glue that binds them together towards these large scale construction projects, maintaining organs and things like that. And so, so we wanted to study that bioelectrically. And so I had two students, Brooke Chernet and Maria Lebikin, who undertook that. And we were able to show that using this bioelectrical imaging, you can tell which cells were going to convert ahead of time. You could also convince perfectly normal cells to become metastatic melanoma just by giving them inappropriate bioelectric cues about their environment. So you can no genetic damage, no carcinogens, no oncogenes, but just the wrong bioelectric information, and they become like metastatic melanoma. And best of all, they were able to show that you can actually reverse carcinogenic stimuli, for example, human oncogenes, by appropriate bioelectric connections to their neighbors. So we had a whole set of papers showing how to control cells bioelectrically. And Juanita Matthews in my lab now is trying to take all those strategies into human cancer. Let's go back to the beginning and ask what cancer is. So I think asking why there's cancer is the wrong question. I think the right question is, why is there ever anything but cancer? So in the normal state, you have a bunch of cells that are all cooperating towards a large scale goal. If that process of cooperation breaks down and you've got a cell that is isolated from that electrical network that lets you remember what the big goal is, you revert back to your unicellular lifestyle. As far as, now think about that border between self and world, right? Normally, when all these cells are connected by gap junctions into an electrical network, they are all one self, right? Meaning that their goals, they have these large tissue level goals and so on. As soon as a cell is disconnected from that, the self is tiny, right? And so at that point, a lot of people model cancer cells as being more selfish and all that. They're not more selfish. They're equally selfish. It's just that their self is smaller. Normally the self is huge. Now they got tiny little cells. Now, what are the goals of tiny little cells? Well, proliferate, right? And migrate to wherever life is good. And that's metastasis. That's proliferation and metastasis. So one thing we found, and people have noticed years ago, that when cells convert to cancer, the first thing they see is they close the gap junctions. And it's a lot like, I think it's a lot like that experiment with the slime mold, where until you close that gap junction, you can't even entertain the idea of leaving the collective, because there is no you at that point, right? You're mind melded with this whole other network. But as soon as the gap junction is closed, now the boundary between you, now the rest of the body is just outside environment to you. You're just a unicellular organism, and the rest of the body's environment. So the idea that what's going on in cancer is a shrinking of the cognitive light cone leads directly to a research program where you say, well, instead of killing these cells with toxic chemotherapies, because I believe that they're genetically irrevocably damaged, maybe what we can do is force them into better electrical networks with their neighbors. And that's exactly what we've done. And so we've had lots of success expressing strong human oncogenes in frog embryo cells, and then leaving the oncoproteins be, but instead making sure that they're connected into tight electrical networks with their neighbors, and they normalize. They make nice skin, nice muscle. They do their normal thing instead of being metastatic. It's interesting because a lot of the interventions we use now, we use drugs and we use materials, screws and bolts and things. And then occasionally we use like a pacemaker or something. But generally our interventions are very low agency, and ultimately we'll have like smart implants that make decisions for you and do, but mostly they're very low agency kinds of things. And when you're using low agency tools, the kinds of things you can do are typically very brittle. In other words, this is why it's hard to discover drugs that work for all patients. You get side effects, you get very differential efficacy in different patients, because you're trying to micromanage at the chemical level a very complex system.
You don't just want agency; you want agency that's attuned to yourself, because
exactly.
You can get someone else's agency.
Oh, yeah.
Yeah. It's not great for you.
Exactly, which is why your own cells coming from your own body, they share with you all the priors about health, disease, stress, cancer. They're part of your own body. They already know all of this. And so part of this is creating these, understanding how to create these agential interventions that can have these positive effects. I mean, we didn't teach the anthrobots to repair neural tissue. We had no idea. This is something they do on their own. Like, who would have ever thought that your tracheal cells, which sit there quietly for decades, if you let them have a little life of their own, they can actually go around and fix neural wounds. Almost every aspect of biomedical needs, all of these things boil down to one fundamental problem. How do we convince a group of cells to build a nice, healthy organ? We may not want to, in fact, we're not going to be able to micromanage all of the gene expression, all of the cells, all the stem cell activity towards complex states that we're trying to achieve. For example, regrow somebody's limb or provide new eyes or normalize a tumor. But if we can take advantage of the native competencies of cells that are already good at solving certain kinds of problems, then we have great ability to achieve large-scale outcomes that are too complex for us to micromanage. So I'll give a simple example. In the frog, which is an animal that normally does not regenerate its legs as an adult, but what we've discovered is that actually we can give those cells a trigger that take them down to the path of building an entire leg. And it's not because we told it how to build a leg or where the leg goes or how many toes the leg is supposed to have. None of that. What we did was to communicate to those cells a simple prompt that causes them to take on the path of going in morphospace, of moving from that injured state to the regenerative state. And so all of the tools of neuroscience, of behavioral science, they all work in other spaces, in morphological spaces and in physiological spaces. And so this is now the opportunity for biomedicine.
a lot of people are really worried that we're going to get supplanted. I think we will, and I think we should. If you think forward, 10,000 years from now, what do you want to be living on this earth? Do you really want it to be the exact same humans that we have now that are subject to lower back pain and stupid infections, and and they have about 80 years of productive lifespan, and then the kind of things like, why is that something that we're attached to? I'm not interested in that at all. I think that any improvements that we can make to enlarge our cognitive light cones, to improve on our cognition, to have better embodiments, whether those are engineered or bioengineered, or I don't care. We can certainly do better than we do now, right? If anybody that's been to a hospital knows that this is not a great embodiment that we're in right now. I mean, it's amazing, but the future should be way better than this.
We produce something called a Xenobot. It's named after a Xenopus laevis is the name of the frog that we use, and bot because we think it's a biorobotics platform. At the simplest level, a Xenobot is what happens when you take some skin off of an early frog embryo. You take it away from all the instructive interactions and all the hacking that normally causes those cells to have this kind of boring two-dimensional life on the outer surface of the tadpole. And you ask them, well, what do you want to do now that you're not being controlled by these other cells? And what they do is self-assemble into this round little thing, which we call a Xenobot, that starts to move around on its own. It rows against the water and against the bottom of the petri dish that they're sitting on, but it also does things like spontaneously turn around. Another example, one of the amazing things that they can do is they do something called kinematic self-replication. So remember that we've made it impossible for these things to reproduce on their own because they're just skin. There's no neurons, none of the rest of the systems that are needed for frog-style reproduction are there. But if you provide them with loose material, so basically loose skin cells in their vicinity, what they do is they run around and they collect those materials into little piles, and they sort of shape the little piles and make a little snowball out of them. That little ball matures into the next generation of Xenobot. And guess what it starts to do? It does exactly the same thing. It runs around collecting more cells and makes the next generation of Xenobot. This is basically von Neumann's dream of a robot that goes around and finds materials in the environment and makes copies of itself. And none of this was obvious at all from what we know about frogs and tadpoles and their genome. And so we're just scratching the surface now of what they're capable of. We're actively researching their ability to sense and remember and what preferences they have and can they learn and all of these things. And so one answer to what is a Xenobot is it's a biorobotics platform where we can take advantage of the assembling properties of frog skin cells to make useful biological robots.